Thank you very much. I am very happy to stand here, and I am very happy to step into the big footsteps of Professor Sorgeloos, who founded our lab, and to present the education and the research, part of the research that we do at our lab. I will start with education. There are two master programs that we offer. One that has been founded by Professor Sorgeloos, the Master of Science in Aquaculture. And in that master program, we focus on 4 themes: feed, farming, health, and life cycle, both of fish, crustaceans, mollusks, and seaweed. And the objective is culturing tasty and safe seafood in a sustainable way. I mentioned health, which is the focus of our second program, which is an Erasmus program, coordinated by our lab, which would also not be possible without Ghent, who is also present here. And this is our International Master of Science in Health Management in Aquaculture, or short, our AquaHA training. There's 4 partners involved, so Ghent University is coordinating, then we have NTNU in Norway, we have Wageningen University in the Netherlands, and UAB in Barcelona. And we also have 3 associate partners, Kasset Sart University in Thailand, Kantö University in Vietnam, and also Terengganu University in Malaysia. The program is set up as follows. So the students start their first semester at Ghent University, where they learn the basics of health management and aquaculture. And then they have 2 semesters that they follow with one of the partners. In Norway, the program is focused on ecosystems and health. In Wageningen, on disease prevention, management, and health. And in Barcelona, on physiology and health. And then in the 4th semester, they have the opportunity to do their thesis with one of the partners or with one of the associate partners. And this brings us to the research that we are doing at the lab. So Jorg will lead you through the lab. This is a lab tour. So we cultivate microalgae, which is mostly to feed the species that we have in the lab. We also have bioflux at very small scale, and in that same lab we currently have a broodstock of European flat oysters to see how they spawn and how the larvae settle on several substrates. We also have freshwater macrobrachium, which is a stock, by the way, that was already from Professor Sorgeloos, so they are doing quite well in our lab, the macrobrachium. Let's not talk about genetic diversity. And then we also have macroalgae, which is mostly by our colleagues of the Ficology group, with whom we collaborate closely together. Here, for example, you see baby palmaria, so different stages of the macroalgae. Then we have saltwater shrimp. We shortly had Caulerpa in the lab. And then, of course, the reason why we are here today, we have Artemia, which the lab also thanks its name to. The research at our lab focuses on 4, well, actually the main theme is aquatic animal and human health, and then we focus on live food and feed, microbial management, and blue growth and sustainable aquaculture. And there's also a 4th circle that you see, new opportunities, to not exclude anything that we think is interesting. So we'll start with live food and feed. Where we have recently, or it's in press, it was already mentioned in the speech of Dr. Matthias Halwert, our Professor Emeritus van Stappen and Professor Sorgeloos, together also with INVE, Geert Rombout, they have written a manual on the production of Artemia. It's in press, so it will be out soon. With a lot of thanks as well to Chris Mailleux, he's our technicus, who has provided a lot of really nice videos to illustrate the manual and also the photos that are in there. We also, together with the VIB, have completely sequenced the genome of Artemia that was published by Stefanie de Vos and team 2 years ago. And then some new research that we are starting up is firstly to grow Artemia on wastewater as alternative proteins. So the main idea is to have microbes, microbial proteins, to feed those to Artemia to enrich them, also with some like lipids that they are lacking, and to then feed or those enriched Artemia to fish, to shrimp, but also to have that as a direct source for humans. And well, not on wastewater, but on byproduct water. We would also like to set up a collaboration with Dr. Jung. Then we also focus on microbial management, and for that the gnotobiotic Artemia model is very important, is central, both for PhD thesis, other theses, and also for company trials who are very interested in the gnotobiotic model. And one of the new researches that we are starting up is to use this gnotobiotic Artemia model to see if insects and extractions and fractionations of insects, such as a black soldier fly, whether they can have an influence on the immunity, on antimicrobial activity, and on cytotoxicity, which is a collaboration together with two other universities in Belgium, KU Leuven and University of Antwerp. Then one of my students, Zhao, who also prepared this for you, is working on alternatives to antibiotics, which is also a central theme in the lab. So she is comparing the survival rate when you add a probiotic, Bacillus, after an infection with pathogenic Vibrio. She tested this in 2 salinities at so normal seawater, the 35 ppt, and also in brackish water, 10 ppt. The food that we gave to the Artemia is autoclaved Aeromonas, so killed bacteria. The probiotic dose is 10 to the 5 cells per milliliter. The pathogen dose was higher. And then the density of Artemia, they were stocked at 20 nuclei per 10 milliliters. And what we saw, and she just sent these results yesterday evening as she is processing the results, is that the control, this is just the pathogen that you give, and these are the Artemia that are not challenged. And on the y-axis, these are the several probiotics that we have given. And this is the survival that is being represented. And here for the first experiment, and here for the second experiment in the high salinity, well, high, 35 ppt. And we see that one of the Bacillus strains, so one of the probiotics, is significantly increasing the survival when compared to the control, which is infected without having been supplemented the probiotic. I must say those Bacillus strains do better in freshwater and in brackish water. So when you see the second experiment, it's not very surprising that here we see that only the 10 ppt, the Bacillus is doing even a better job, and the survival is not significantly different with the unchallenged one, but it is significantly different with the animals that are being infected without the Bacillus. So this means that we have a probiotic that's working quite well against a very notorious pathogen, Vibrio. Another way to strive for alternatives for antibiotics is to look into phytobiotics, essential oils, herbs, and I'm very happy that we had very good discussions with UMT this week to see how we could do a joint PhD on this topic, but also with CityU in Hong Kong and with the South China Sea Fisheries Research Institute in China. We are looking at joint collaborations for this topic. Then something maybe not related to Artemia, we also test different alternatives to animal models, in which, for example, we have a skin explant model. So basically you take out a part of the skin of the fish, and this is in a 3D printed model. So here you can add the physiological, so the seawater, for example, the exterior of the skin. You can add chemicals, pathogens, and see what influence it has on the skin, which is squeezed in between the two parts. And then here you can add the physiological fluid, which mimics the inside body fluid. We have the same kind of model for the gills, so in which you take out the gills and you connect the gills to a perfusion fluid, and then again you can put those in a tray and add some chemicals, add some bacteria, and see what influence it has on the health of the gills. We also are quite good at histopathology. We do this in collaboration with the Faculty of Veterinary Medicine at Ghent University. We of course have our luminescent Vibrio, which only luminescence when it is pathogenic, so when it can kill your fish, your shrimp, that it will infect. And when it's less luminescent, then it means that the virulence is decreased, which is a very nice tool to test if you have some chemicals or some probiotics that work against the virulence. So this is one of the techniques that we use to see is something virulent, yes or no. And then together with the Faculty of Veterinary Medicine, we can do histology, scanning electron microscopy, and transmission electron microscopy to look at the cellular level, whether a tissue is affected or not, which of course also works for Artemia. Then we also have a 3D model. This is now the example of a mussel, but we have also done this with Artemia. So basically you start from your host, which in this case is Mytilus edelis, a mussel. Then you make histologic slides and you stain those. And then you transfer them into gray values and you select or you identify each of the organs in those histologic slides. And then up next, you can make a reconstruction via software and then you get a 3D image. This of the inside of the mussel, but as I said, we have also done this for Artemia. And this is in collaboration with Professor Wim van den Broek and Professor Pieter Corneli of the Faculty of Veterinary Medicine. Then we also are working on blue growth and sustainable aquaculture, which is very important in Belgium. So this is basically our offshore research that we do. We cultivate oysters and macroalgae and also restore the European flat oyster in the Belgian part of the North Sea because it has a high societal interest, a huge valorization interest, and then I mean from the higher level, from our minister and also from the rector of UGent. So here you can see how we installed seaweed nets. Here are oyster structures. And these are cages with oysters inside for restoration. And this all in a wind farm, which is the first time that this has been done in a wind farm in Europe. Last but not least, we also have new opportunities, and this is my student Chang who is working on this, to see how aquaculture can serve as a source of blue biotechnology. And we determine whether the collagen that you can extract from trout skin or from another fish skin, in our case it's rainbow trout because we cultivate that in the south of Belgium, and alginate extracted from sargassum, which is growing in Ostend in Belgium at the coast, how those two can be used in a hydrogel, which you can see here, for wound dressing, both for veterinary and human medicine. And the first results are very promising. So the combination of these two in a gel is giving as good results for now in vitro, so not tested on patients yet, as compared to commercial hydrogels that are currently on the market. This is in collaboration with Professor Peter Dubruel and also with Professor Warte Spiegelaar, who is of the Faculty of Veterinary Medicine. And I want to thank you. By having said that, I would like to welcome you all at the Larvae 2024 in Ostend in Belgium. And thank you very much for your attention.
Thank you, Prof.